All right. So welcome everyone. Great to see you. See you all joining, and they're going to be filtering in at least for the first few minutes of this session. It's a pleasure to have you all. Thank you for the response to this series. Um, I would say that this was an idea put forth by Saurabh and Deepa to India Bioscience, and we were happy to get on board. The whole idea was to bring leading scientists to India virtually, at least, and so that they could share their contemporary research with the life science community in our country. So thank you, Deepa and Saurabh, for the idea. And India Bioscience is very happy to get on board and facilitate this towards our larger science community in India. We have a very exciting and interesting guest scientist today. And we're going to keep this series going in the next few months. So look out for new announcements. But for now, I will ask Saurabh to introduce our guest scientist, share a few housekeeping rules, and look forward to the talk.
Welcome everyone, and this is absolutely exciting because we received a massive response, like almost more than 400 participants, and they will keep joining. And so to cut it short, actually, I'm not going to give Alfonso's introduction very formal with CV and the awards he has received over the years, but it is like how I got to know him first time was through the book, Developmental Biology book. We all read probably the Principles of Development, where he contributed immensely. And then later on, he has also written 2 more books on the similar subjects. He was a professor at Cambridge University in Department of Genetics for many years, and recently, like few years back, he moved back to Spain, his home country, and joined University of Pompeu Fabra, where he is continuing his research, interesting research. One of the very fond memory of me was when I was visiting Cambridge. I was studying gas relation in my PhD, and it was very exciting to discuss with him, as you can imagine, like discussing the gas relation with the person who has contributed so much into it. And without wasting more time, I would like to invite Alfonso to start his talk. And the few house rules is like you can start putting your questions. And since we have many participants, if we cannot take all the questions, please send it to us and we will try to get back to Alfonso and get back to you with the answers if possible. Yeah, Alfonso, please.
Thank you, Saurabh. It's very, very embarrassing. And you've set up a challenge here and you've set up the audience to be disappointed. But, we'll do what we can together. If anything goes well, it's the success of India Biosciences. And I hope I can tell you some things that you can learn from. When I was proposed to me to do this, I was asked to do what I've tried to do, which is a mixture of how I developed my career and how I developed my science. And as you know, for us scientists, there is not a big difference between our lives and our job. That's something that surprises some people, but that's the way it is. I decided to call the talk Making Choices because, as you will see, all my life I've been interested in how cells make choices. But when I look back, and one of the things that this talk does is allow me to go back in time, it makes me think that my life has been in parallel about studying how cells make choices and how did I make choices when I look back. I should also say that this talk has made me feel like a time traveler. You all know this film, and I do feel that I come not from the future, but from the past. I think when I look at the world today, and this talk has made me compare it with what the kind of science I grew up with, I can see that it's very, very different. Some of you sometimes look for advice on people like me, and I will try to give you some thoughts at the end. But what you have to understand when you listen to people my age or of my generation is that the circumstances that we lived through when we were your ages and the challenges that we faced were very, very different. I don't say anything that you don't know if I tell you that things were easier. the environment, the social, the political, the scientific environment was easier. There was less people doing science. There were big questions around. There are big questions around today, but they are hidden. But I will leave that from the end. I just want you to understand that you're talking to a time traveler from the past. So I started my career long ago, as they say in the movies, in a country that is Spain, that you can see where it's there, and you can see where I studied in the Universitat Complutense of Madrid. That's where I did my undergraduate. In a trip last year, they had my card, and that's my card from the genetics department where I studied. So I found it amusing, and that's where you are. This thing here, I don't have a pointer. I'm going to try to get a pointer. Laser, I don't find it. Don't worry. On the top bottom, it says notable. That means B. I was always a B student. I was never an A student. I oscillated between a lot of things. Now, my years at the university, those 5 years were really very difficult. And the reason for that was this. I grew up in a dictatorship, and these were the last days of that dictatorship. And what happened is that most of the time the lectures were being invited by workers asking us to support their rights. And we shared a lot of the learning with the police, who kept that very closely. Sometimes I'm just laughing because it was very easy to see the secret policemen because they were the only ones wearing suits and ties in the lectures in the university. It was very difficult to learn from the classes. However, you could read. And I always found intriguing that although the regime sort of banned a lot of interesting books of literature, they let other books to read. And the first book that really had a big impact on me is this one, which is by Jacques Monod. Jacques Monod, I hope some of you remember him from Monod and Jacob. Those are the ones that really lay down the foundations for our understanding of gene regulation today. That book, it's called Chance and Necessity. I reread it over the pandemic and found it a bit dated, but at the time it was profoundly life-changing for me, because all of a sudden I could see that there was a lot in biology, particularly in a very reductionistic approach to biology, that made it very attractive to me. Another book that I found in that time was this one, which is called, I put it the books as I have them in my library still these days. What that means is towards a theoretical biology. And you will see at the top a name that is very familiar to a lot of you, which is Conrad Waddington. Waddington was a very interesting person. Many of you know him because of the Waddington landscape. He was a person that was very much ahead of his time, thinking a lot, not only doing experiments. He was the first one, for example, in proving that organizers existed in mammals and in birds, something that had been shown in frogs. But he also had visions of biology and had an interest on bridging theory and experiments in biology. And he had a number of conferences in Italy, which he then put in the form of a book called Towards a Theoretical Biology. For someone like me, in particular, who had spent all these years embedded in a rather pre-revolutionary environment with policemen, just reading, with very little ability of doing practical biology, the idea that you could do theoretical biology was actually very appealing. Particularly, and this was my first encounter with what Saurabh has mentioned, Louis Wolpert, it came to me through this book. And what that book contained was one of the chapters by somebody called Louis Wolpert on positional information and pattern formation. That's what that means in Spanish. And in the article, it says, our almost total ignorance of the mechanisms associated with pattern formation in animals is a serious gap in the current theory of biology. That really grabbed my mind. And I thought that you could actually do theory in biology. The problem that really interested me, particularly after reading that book, was how development proceeded. There was some kind of magic in the appearance of structures at the end of development in different organisms, particularly in human beings, and bringing together these ideas that I had gathered from reading Monod and the ideas of Waddington. I thought that one should be able to put forward some theoretical components and bring them together into some global theory of biology from the microscopic to the macroscopic. The same way that physicists can predict all these possibilities of the universe from the initial particles, I thought this should be able to do with biology. Just behold what naivete does, okay? This is really, when I look back into it, this is about mid-1970s. I think I was very naive. But sometimes when you are bored and when you think, well, you can aim high. I was very, in the final years of my university, I took a lot of courses in chemistry and in physical chemistry. And this idea that you might be able to do theory in biology was very much in my mind. So I was lucky enough to get a Fulbright scholarship, and I went to the University of Chicago, where there was a program on biophysics. And there I started trying to think about the ideas, how to develop the ideas that I want, and to try to do a thesis, thinking that biology would be a matter of genes, cells, and numbers. This is really what I was. I hope we are still on. This is almost. I'm just going to check the chat. That okay, looks like we are on, because sometimes you know we get. I just didn't want to be disconnected and talking to myself. Now, what you can see is that this was the idea. These books by Ilya Prigogine made a lot of impact on me. This was the first people to actually develop theories from thermodynamics in biology that really mattered. And that really, the first few years, I was very interested in this. And particularly one thing that I learned was this branch of physics called statistical mechanics, whereby you can predict the properties of the gases at the macroscopic level from the particles at the microscopic level. This gentleman there is Ludwig Boltzmann, who has this formula that many of you, all of you, will have seen it in school. And I liken this idea of statistical mechanics explaining the macroscopic properties from the microscopic properties to the problem that we had in biology, which is how to explain these marvelous phenotypes that we see in tissues and organs assembled into organism from the activity of genes and proteins. I don't need to tell you that problem is still languishing. Now, one thing that became clear in my first 2 years in Chicago is that I was no mathematician. I think those years influenced me a great deal, as you will see, and they left something lingering in my mind, but I realized that I was no mathematician. And in many ways, I think what happened is, in the next slide, that I ended up doing a PhD in what was fashionable at the time. So this was a lesson that I look back, that my first 2 years there were a really train crash. I mean, it was very, very difficult. I had to accept that I wasn't good about what I wanted to do, and that biology was not ready for that kind of work. But importantly, I could not do that kind of work. A very kind head of department suggested that there was a number of techniques that were coming into the frame, and that was molecular genetics, and maybe I would like to do molecular genetics with someone that had just arrived to the University of Chicago. The man there is Malcolm Kesadavan, who was one of the pioneers of molecular genetics, and that's part of my thesis. I ended up doing gene fusions using beta-galactosidase. In the meantime, I kept on reading developmental biology, something that infuriated my advisor, who every time he approached my desk, what he found was not many papers on molecular genetics, but lots of papers on developmental biology. And the one thing that happened in 1983, which really determined a great deal of my development later, was the appearance of this paper, now classic in Nature, from Nusslein, Vollhart, and Wieschaus, although the screen was also done with Gerd Jürgens, in which they had done a saturationist mutagenesis in Drosophila for mutants affecting pattern formation. And as many of you know, this screen revealed and opened up a lot of the modern molecular developmental biology. A visiting lecturer, an Italian visiting lecturer, alerted me to things happening in this field and suggested that I went to Cambridge as a postdoc to work with Peter Lawrence, because some things were going to happen in this field. At the moment, what Nusslein, Vollhart, Wieschaus, and Jürgens had done was to reveal the mutants. This may have been the key, that maybe the key was there waiting for the getting into the mechanisms that were behind that. So I put behind the idea of doing theory in biology, and I decided to be an experimentalist. And in 1983, I moved to Cambridge, to the old Laboratory of Molecular Biology, to work with flies, working with Peter Lawrence there in the middle. But my real mentor at the time was Michael Acham, and he's been a real mentor all my life. Lesson for students sitting in the background and listening in the background. I think whenever you are doing your PhD or you're doing your postdoc, your supervisor, it's all right, but you will always need someone outside to advise you, to guide you, to make you think about things that your supervisor might not give you. Might be that your supervisor is great, and I'm not saying that Peter wasn't, although we argued too much, but it's always very good to have someone outside, and I considered Michael Acham my real supervisor, and that supervision has continued throughout life. So I was, for those 4 years, in the laboratory of molecular biology, and although I was a postdoc with Peter, one of the good things of the laboratory of molecular biology, I don't know that it's still the case, is that really there were not labs, and I really have to be grateful to Peter because he let me do whatever I wanted to do. And what I wanted to do very quickly was to start working with Michael Acham, and I was very lucky to participate with Michael in this paper here, which is a set of in situ hybridization of the gene ultrabithorax, one of the Hox genes. In fact, this is the first one of the first in situ, certainly the first one with great detail of the gene ultrabithorax in a Drosophila embryo. You can see on the right that one of the things that appealed to me working with Michael was his interest in quantitative analysis. And I cannot tell you how many hours of inputting grains of tritium with an elaborate device that Michael had set up with an old Amstrad computer to get those patterns of expression of different probes of ultrabithorax. I'm going to stop because on the one hand, I get emotional. On the other hand, I could go on the detail when I look back at this. This was quantitative biology very, very early on. And I would say that this is one of the 2 or 3 papers that if you ask me, what are the pieces of work that you are most proud of your life? This is one of them. And I'm very grateful to Michael for having allowed me to be part of this work. The other thing that I did, particularly at the LMB, was to start using those mutants that were from the screen of Janni Nusslein and Erik Wischhaus to try to understand pattern formation. And I focused on the so-called segment polarity genes, which affected the pattern in every segment. What you can see there on the left is the larva of a Drosophila melanogaster with all the segments, and the segments, you can see that they have very precise patterns. At the bottom on the right next to it, what you have is a wingless mutant. Wingless is the originator of the Wing series that is very famous. You can also see at the top the expression of wingless in blue and in Grail in red, and what happens to the pattern formation. Studying these mutants and how that pattern of stripes arises led us to the paper that is indicated there, which started to tell us how cell interactions between cells, generated those patterns. This is the way we worked out at a very high level, which later has turned out to be true, how genes that are now very familiar, like hedgehog, like wingless, like engrail, like patch, they are genes. We used the genetics at the time to work out how they interacted between cells. I mean, that figure is from the paper, and I'm very, very glad that is worked out. But this study also marked my life because it made me see, as it's independent here, that against what I thought at the beginning when I went to Chicago, that biology, it's a matter of genes, cells, and numbers. But above all, it's a matter of genes. And as I was going to discover slowly, it's a matter of cells. And I do think that that's an important thing, which it took at the time some more appreciation, because for many years we focus our studies on transcription factors, and it's taken a little bit to start appreciating, as I might come back at the end, that what matters is cells. So in 1987, I moved from the LMB, which is outside Cambridge, to the University of Cambridge, where I was going to spend the next 30 plus years, first in the Department of Zoology on the left and right on the Department of Genetics. Cambridge is a very idyllic place, and I'm very, very grateful to have spent and being able to work and live there for all these years. Now there immediately we face, and this is really, I became very interested in wingless and particularly, and what it's important is to understand the context of development. I cannot tell you all the details. I'll give you the summary of what 10 years of work led me to looking at the fly. And really, when you look at development, this is an outline of the development of a vertebrate, for example. What you can see is a series of branching patterns. It's over time. When we put vertically time, you can see that the different tissues appear through this branching process. And in these branching processes, if you look in detail, there are always binary decisions. The cells are making decisions. So it's not only we that make decisions, cells make decisions, and probably in the end, there is not much difference between the way cells make decisions and the way we make decisions, with one important difference. Probably we are irrational and cells are rational. And this is something that we are trying to find out. That rationality says that they might be patterns, and thinking a lot, I wonder whether really when you look at those diagrams, what they might be reflecting is that at every decision, a cell is deciding between two fates. And this is something that is really marked my life. The fates could be different cells, or it could be remaining like the starting cells and moving on. But I do feel that this is one of those principles that arise in biology. And I'll be happy to answer questions if any of you think that there is more to it or that I have missed something. And we can see this in a very, very classical example in biology, which is C. elegans. C. elegans, as you all know, follows a stereotype pattern of cell divisions, and at every cell division, cells make a choice about their fates, and really the organism appears through a sequence of spatially organized binary cell fate decisions with great precision. So we do know how this happens, and we've learned that really cells are throwing a coin, if you wish, to the air, and that's what lives, and there is a certain stochasticity, to use that word that has become very, very famous, in their choice. There is a chordate which is very similar to C. elegans, and is the ascidians, which you can see below. It's a chordate, it's a relative of ours, and it also makes its decisions in a binary manner. However, we, mammals, vertebrates in general, are very, very different. You know, the bet was that there was also binary decisions, but the decisions were not undertaken by individual cells, but by group of cells. And what you had was sort of many coins thrown into the air, and then some of them would become heads and some of them would become tails. The problem for us to find out, I mean, I could prove that very easily. I'll give you some ideas in a minute, but what is the big problem that I don't think has been solved at all, it's very difficult to tackle, is that the proportions of head to tails are always different from one tissue to another, but sustained from one tissue to another. And I don't think we know how cells are able to measure those numbers and reproduce. And this is a question of cells, not only of genes. The two mutations in Drosophila that really got me thinking, and with which I worked for many years in the first phase of Cambridge, was wingless and notch. And this is a very good moment to highlight that the allele of wingless that you have there is a viable allele. The mutation that I showed before is a null allele that leads to embryonic lethal. This allele, which is wingless 1, was found by Sharma and Chopra in India. In fact, wingless is a mutation that is discovered and comes to the world via India. And this is something that we should not forget. And the mutants that Sharma and Chopra found don't look very, very different from the one that you can see there, which is lacking a wing on one side. It is more subtle than that because it's a transformation of the wing into the notum. And it was work with these two mutants and a lot of genetics in many different systems within the fly that led to a certain view of fate decisions, which has colored my life for the last 20, 25 years. And the proposition is the following. What you have is that when a cell makes a final step of differentiation, what we notice is that they go through a state, which is when the cells are throwing the dice or the coins or whatever you want. So the cells are thrown into a state in which they have to decide between going forward and differentiated and going backwards. This is where the proportions are decided in this particular moment. I call that state a transition state and suggested that it is an intermediate of cell fate decisions in which a cell has a mixed identity. So what we suggested, and as I say, this all comes from years of looking at fate decisions in Drosophila, is that the cells can have a cell can have two identities at the moment. I mean, think about when you're making a decision to eat in a restaurant or to choose your girlfriend or your boyfriend. Sometimes you have different possibilities in your head at the same time, and you're choosing between one. Cells are doing the same thing. That means that it is characterized by heterogeneous gene expression. So those cells are expressing genes from two different fates. It is reversible. The transition state is reversible. The decision is irreversible. And we suggested that it is universal. So you can have the situation there. The transition state is in the middle. And what genetics suggested is that the outcome of that decision is determined by balance between notch and wing signaling. I absolutely believe that this is true. Just now, we are starting to be able to go back to this question, but that's another thing that Goethe said, that life is short and art is long, and this is one of the problems, that sometimes if you find a good question, you might not be able to answer it in the course of your lifetime or be sure about it, but it is important to have that question, and in many ways, there is the question that has determined my life for the past many years. On the other hand, as a very good friend of mine once told me, when I said to him, well, I'd like to know if I am right. And he answered to me, how boring. And in a way, it is true. If you know that you're right or you're wrong, your quest is finished. And it is in the uncertainty and in the will to know that a lot of our profession thrives on and feeds on. So there you are. Somehow, something biases that coin throwing, and that's really what I think it's about nudge and wind. This is not just in the fly. I mean, this is a summary of everything I've shown. The literature is full. You probably all hear about wind being involved in everything, and I think one day somebody will answer why, and maybe it will read our papers. But actually, this is very common, that when cells are making a decision during development, one of the fates is driven by wind signaling, and the opposite fate is driven by nudge. We are now working currently in trying to update this work, which, as you can see here, has been without updating. It's a theoretical work, if you wish. It's about collecting information. We also work out the mechanism of this. This was in the fly, particularly the work of Silvia Muñoz Descalzo, now in the Canary Islands. We work out the mechanism. Nobody pays too much attention to this, probably because it is difficult and we are too engaged with epigenetics. But I know that everything that we said there, it's absolutely true. And we actually, we focus a lot of the work in wind to the beginning to understand one side. In a way, notch is a way to regulate the wind decision. And that is what we have been working. This is a review which is often cited, in which we suggested that cell-fate decisions in general in development are noisy. And the role of something like wind is to denoise the transitions. And this is, I mean, I think there is increasing evidence for the noisiness, for these heterogeneities in gene expression that you hear about very often these days are reflecting the heterogeneities that arise when the cells are trying to make decisions. And that's been very important. Now, that was what we concluded. On the basis of existing evidence, we proposed that the denoising mechanism depends on the wind signaling pathway. I believe in this, and we continue to work on this in a very slow pace. A very important moment in all this job and in trying to put my thoughts and the framework in a clear sort of frame came with an invitation from the EMBL cell biology unit run by Eric Carcenti at the time to a retreat in the trail in Provence. It's a place where people organize small meetings to think about. And there I came across a talk by a then young Michael Elovitz. And this talk completely blew me over. What you can see there is bacteria. They contain a gene green, a gene red. It's the same gene, but with two promoters. And what you can see is those cells, that they have a lot of heterogeneity, depending on whether they are green, they are red, or they are yellow. In fact, what this work showed, it introduces the term molecular noise or transcriptional noise. But you can see that usually the cells are very heterogeneous because they are expressing the same promoter drives expression in different levels, and that is regulated. That's what you can see here with these two. This is a IPTG, is an inducer of the promoters that abolishes the heterogeneities. But if you apply IPTG now in the context, in this case of a REC-A mutant, you recover the heterogeneities. What it's told me is that heterogeneities, as we suspected from the work in Drosophila, are very generous, very general, and that they were regulated genetically. And when I came back to Cambridge from this meeting, I suggested that we ought to work on this, to find a system to work on this in the lab. This is a poem of T.S. Eliot, which some of you know, the Four Quartets. This is Little Gidding, is the last one. And this is a very famous line for that poem. We shall not cease from exploration, and at the end of all our exploring will be to arrive where we started and know the place for the first time. Because those heterogeneities, the ability to quantify them, the ability to devise theories, like Michael Elovitz did and has been doing since, it's basically the thing I was interested 30 years earlier when I went into Chicago. It was very, very, very late for me, but I'm very glad. Some of you might know that I organized this series of conferences in Cambridge, the physics of living matter, for 13 years. I'm very glad to see that it keeps going. This year is the 18th celebration. And it's been a pleasure to see the development of a true biological physics. Today, nobody questions that physics is a part of biology and is really revealing a lot of very interesting new vistas of life. And there it is, the cells here in this middle are like throwing the dice, and then you have exactly the same situation depending on the regulation. And as far as I'm concerned, this is not very, very different from the way I was thinking about life. Now, upon this, I mean, one might think, because that's driven genetically, that then the development of an organism is just a mapping of gene space into cells. I mean, there is many interesting observations about that. And as many of you know, this is pretty much the moment in which we are. I mean, we seem to spend our life doing single-cell RNA-seq because reviewers ask us for it and editors plea on us to do it so that we can publish our papers. And the feeling, sort of the idea is sitting around that really, if you know the single-cell RNA expression of a group of cells, you know what they are doing and you know who they are. However, I think, as I've hinted, and really this has always been the underlying leitmotif of my career, is that the development of an organism is also, and maybe not just also, is first and foremost about mapping cells into tissues and organs. And you can see that wherever you look, what you see is not genes, you actually see cells. We are told that the genome is a toolbox, that the genome is a blueprint for the organism, but nobody tells us who is using that toolbox and how. And the real uses of the toolbox, the structures or the beings that actually use that blueprint and build something are cells. And I will claim that the same way that the 20th century has been the century of the gene, the 20th century is the century, the 21st century is the century of the cell. More about that in a minute. So if you think about it, what DNA is, it contains instructions to form nanomachines. Nanomachines in this context of cells, which are entities that are very ancient and that cannot be reconstructed from DNA alone, once those nanomachines are in the context of cells, those nanomachines acquire new properties that feedback on the DNA, that feedback on themselves, that creates new behaviors. And the cells, using all those new behaviors, create tissues and organs. The cells are at the center of any developmental process. Probably they are at the center of life. And nowhere is this more beautifully displayed than in the development of different organisms. There you have some familiar to some of you. On the left, a fly, on the top, a fish, at the bottom, a chick. And development is just very, very aesthetically pleasing. And that is not the action of genes. That is the actions of cells controlling genes that are downstream from them and building these beautiful structures. Now, at this point, I had to make a choice. We were working in Drosophila, and I had to make a choice, and I was very excited by Elovitz's talks. I'm having read Elovitz's work, and I had to make a choice of paths. I could have continued to work that in Drosophila, or I could move to some other system. And at the time, I heard about embryonic stem cells. This was very early. This was about the early 2000s. And I decided that to bet that these cells would be a very good system to study development. At the time, people were starting to talk about embryonic stem cells in the context of curing diseases, and they still are. But I thought also that they would be a fantastic system to study development. This was a very difficult choice because we hadn't worked with even culture cells before, and it was not easy. But I think we were very, very grateful to a lot of help that we got. We were extremely lucky that at the time that I decided that we should go into this, Austin Smith had moved from Edinburgh to Cambridge, and his longtime collaborator, Jenny Nichols, were both extremely kind to us in teaching us how to deal with these cells. I do feel that our lab has very high standards, not because we are great, but because Austin and Jenny were incredibly good and incredibly generous with everything and with all their knowledge. Now, just to remind you, embryonic stem cells are cells derived from very early mammalian embryos that can be cultured indefinitely in the dish, and that whenever you put them back into an embryo, they go and will contribute to that embryo, no matter how long they've been sitting around. In vitro, you can coax them into differentiating into the different cells of an organism, and but they will not make an organism when you have them in culture. One thing that appealed to me that harks back to a slide that we had before is that the way you make a mammal is very different from the way you make a fly. In fact, it's easier. A mammalian embryo is an easier entity than a fly, because in a fly, you're making everything at the same time and in a great hurry. And that will sort of sound familiar to those of you who work with flies. Whereas a mouse is slow, it's actually boring, and it's a sequence of binary cell fate decisions. So it has many advantages. If you want to learn how cells make choices, you want exactly a system that is slow, where every decision takes time, and with just a sequence of fate decisions. All these were very appealing to me. There was one interesting question in this system. Why was it that you could not get cells to make structures like embryos in vitro? We started by working here. We had a very lucky collaboration with Jordi García-Ojalvo, who has been very important in convincing me to move back to Barcelona. He's a theoretical physicist who works with Michael Lelovitz. This is the way I actually met him, through his papers. And we had a very good series of papers on modeling and quantifying a lot of the properties of the embryonic stem cells. And this was interesting because this allowed me to bridge my interest from when I went to Chicago with the interest that I had been developing over the years. However, development is not about genes. I started, this was good, as I said, because it allowed me to bridge those two problems that I had, but I very quickly understood that really we were dealing with cells. And that while we can model many aspects of gene regulatory networks, in order to understand how cells build organisms and build tissues and organs, we needed a very different way of doing things. This is a very famous sentence. The gentleman over there is Richard Feynman. And what you are seeing there is his blackboard when he died, and he had a number of mottos. You can see there, know how to solve every problem that has been solved. Well, that's not very difficult in biology, because not many has been solved, so that's okay. But the one that really challenges us is, what I cannot create, I do not understand. If you think about it, a lot of our knowledge of DNA is very profound these days, and that allows us to build engineering devices that are helping our understanding of our health and curing health. CRISPR is a very good example. All that comes from a very deep understanding of how DNA works. However, as I have said, that doesn't help us understand, unless you believe that single-cell RNA-seq really leads you somewhere, that doesn't help us understand how cells, the entities that are above DNA and RNA, how do they build organisms? So you can think that if you understand that process, you should be able to recreate it, and this is what we started to do. This is what led us to this system called gastrulates, in which we found that by taking small numbers, very defined numbers of mouse embryonic stem cells, and putting them under very special conditions, after a little bit of time, the little protocol that has become simple, we get these structures which don't look like embryos, but they are very reproducible. And when we dig into them, we can see that they have many, many, many of the aspects of embryos. And there they are. Sorry about that. Now, here you have what happens when these structures, the cells come, they aggregate, and there you have now two movies. The one on the center, it's one that is carrying a reporter for SOC17. It's a gene that produces endoderm, and you can see that the cells will break symmetry and they will have endoderm only on one side. In fact, this movie is one of the first movies that we had. Both movies are the first one. The one on the right is brachyury, this gene which signals gastrulation. And in both cases, what you can see that after a while, what you get is a polarized gene expression. This was very, very exciting to us. And we call these structures gastroloids because when we dived into them and tried to see what is that they were holding and what they were doing, we could see that they imitated, they were a very good representative of the process of gastrulation, of the outcome of gastrulation. Here you have, I thought that this year it was quite funny. Indeed, the first account of gastrolytes I found in my notebooks. As a PI, I thought that a very good way of running a lab would be to keep a diary, and I did that for the years in Cambridge. And you can see that on Friday, the 24th of May of 2013, I did point out that something had happened in the lab that was exciting, and that I would call those things gastrolytes. The one on the right, you can see actually the first gastrolytes, and that led to a paper where Suzanne van den Brink and David Turner were very, very important in developing those structures. I just found it amusing that it's been 10 years since this happened. Time, as you will discover, flies. Here you have, some of you might have seen this, some of these structures. You can see that they have a very nice anteroposterior polarity. Most surprisingly, out of nowhere, you get the 3 axes. You can see that in the right. You can see they have a dorsoventral axis, and they also have an anteroposterior axis, and they are able to do even left-right. And when we look at the genes, we can see that those genes are organized in a manner that is very similar to the embryo, and they follow those 3 axes, and that in a temporal fashion, they also do that. Another important lesson from all this work, which to me was very, very important, I think the first paper we wrote, and that was very important, as I said, the work of Suzanne van den Brink and David Turner. I was very worried about those structures because we didn't have at the time, the way the lab was set up and the funding that I had, we didn't have enough resources to investigate as much as we can. And it happened at the time, I reached out to somebody called Matthias Lutolf, who many of you will know. He's a fantastic engineer. And in a visit to him, I visited an old friend of mine, or an old acquaintance at the time, and Denis Duboul, which many of you know, and I showed him some movies of our gastrolates. He became very interested because for many years he had been looking for an in vitro system to study the regulation of Hox genes. And with these interactions with Matthias and Denis, we actually got great insights into the gastrolates that I don't think they would have happened if it hadn't been for that collaboration. And what is important of that collaboration, as of others that I've had on gastrolates since, is that they were very generous and everybody has pushed in the same direction. In general, I find this a very good property of the field. So here you can see the Hox genes. This is a lot of you there. You have a gastrolate on the top right, sort of elongating. And what's happening during those elongations is that the Hox genes are being unraveled in exactly the same manner that they do in the embryo. And this is what has got the knee going into this. And not only it's a temporal pattern that mimics the temporal patterns in the embryo, but the spatial patterns are also the same. And if you haven't seen it, I suggest that you go to Nature Cell Biology and look at the fantastic paper of Denis on the timing mechanism associated with the onset of expression of these genes using gastrolates to study. So they are very, very useful as tools to study development. So over the last few years, there is many work. I'm not going to bore you with it. And we've been able to find in playing with the gastroloids, the gut, the heart, the blood system. We can get the spinal cord. We can get somites. There is a lot of work that has been going on, that is going, and we are having fun. This is the important thing. Now, embryonic stem cells are like children. They need a lot of pamper, a lot of care. But if you're good to them, they're good to you, and you can learn a lot and have fun in the process. Now, you will have noticed that I mentioned that those structures do not look like embryos. And this is something that was of great interest to us, because although they don't look like embryos, the organization of gene expression, not only qualitatively, but also quantitatively in a space, is very similar to that of embryos. It is as if they are able to reproduce gene regulatory networks without morphogenesis. And what this suggested is that there are two systems working in embryos, gene regulatory networks and what we have decided to call cell regulatory networks. Gene regulatory networks create time, and they depend on the interactions between signals and genes. And I would suggest that cell regulatory networks have to do with mechanics and geometry. In fact, when we start playing with the boundary conditions and the mechanics and the geometry of the gastrolate, we start getting some morphogenesis. So development is really about the interaction between these two systems. And I would say that a lot of the important questions for you guys for the next 20 years is to find out what those connections are and how they work. At this, I really like this statement from Pavel Tomanchak in a discussion on these issues in Twitter, where he says, I find it strangely liberating to think of genomes and emergent cell behaviors as two rather independent entities that need to remain compatible with each other in order to create a functional living system. I repeat, the 20th century has been the century of the gene, and this is great. And we have learned a great deal, and it's put us in a very good position to understand a very important aspect of life. The next aspect of life depends on understanding what cells are and how they control the gene regulatory networks. It means to understand the interaction that is outlined in that slide between gene regulatory networks and cell regulatory networks. Now, I think this is pretty much everything I have to tell you, and I will answer your questions. This is a wonderful statement from a very, very famous mathematician called David Hilbert in a book which I bought it and I didn't understand from page 2, but at the beginning in the introduction, he makes this statement, which I find very, very profound and appealing. He says, it is difficult and often impossible to judge the value of a problem correctly in advance. Don't forget that what we are doing, what our job is to find problems and solve them. We don't just go to the lab to play with genes and cells so that our PIs write papers. We have to find the problems, what I like to call our Moby Dicks, and chase them. So it is indeed difficult and often impossible to judge the value of what you did correctly in advance, because the final award depends upon the gain which science obtains from the problem. And sometimes you might choose something that doesn't mean anything. It is important to you. It behooves us to pick up that quickly. And if you're really interested, find the right perspective. Nevertheless, we can ask whether there are general criteria which mark a good problem. He's talking about mathematical problems that I would say that this is general. A problem should be difficult in order to entice us, yet not completely inaccessible, because otherwise it will make fun of our efforts. That's the essence of science. It should be to us a guidepost on the mazy paths of the hidden truths, and ultimately a reminder of our pleasure in the successful solution. I hope that you can think all that. This I didn't realize, but this has always been underlying the decisions that I have made in my life. Now, a few months ago, I moved to Barcelona, and I'm very happy to have been able to recreate a group here with these wonderful people that you see. Barcelona also has a lot of pleasures, as you can see there, which I had forgotten in England, and it has also the pressures, although Messi now is not in Barcelona, but that's a different matter. Now, there is Barcelona. So, you know, if you ask me, what is the simple advices that you can give us, apart from what David Hilbert suggested in that sentence? Well, read, change, and adapt. This is very important in life, but also in the science. Never stop learning, and certainly never lose the will to know. And don't forget, because this is very, very important, particularly these days, as I will show you in a minute, that science is about seeing, not about being seen. And those are choices that we face. Now, we live in a strange world. We live in a very, very strange world. And it's difficult. It's difficult because, you know, slogans are winning votes. And as that, I read that sentence from a columnist in The Guardian over the weekend, Andrew Rawlsley, about on the one side are bad actors who seek to advance their interests and their causes by peddling misinformation, mendacity, and fakery. And resisting them are those who prize facts, veracity, and rules. Science is about facts, veracity, and rules. And we do have to remind ourselves that no matter how appealing it is to get a headline in a newspaper or to get something. And I say this in the context of much that has happened last week in our field, which is summarized here. Last week, The Guardian threw that tremendous headline, which actually it happens not to be true, but it's thrown the field into great disarray. And the lady in the picture on the left seems to have got the eye of The Guardian. Remember the face of Andrew Rawlsley. If we cannot allow that, there is no proof of anything apart from the use of the terms. And I suggest to you, because this field belongs to you, the future belongs to you, we cannot allow science to lose some of its most important elements. I told you that we have to adapt to change. I'm not going to tell you that if you follow the path that I have followed, you're going to get success, because we are in a very different environment. But we have to adapt what is the essence of the science to the modern times, and not get the modern times to determine how we do a science. Science has checks and balances, and we have to make sure that those checks and balances remain in place. It's important to talk to the press, but when we talk to the press, we should make sure that the statements that we make reflect the findings that we have. I'll be happy to answer questions on this. And so I take this opportunity. This is the first time I thought I wasn't going to do it, but actually I spent the last few years writing this book, which is really a summary of many things in my life in Cambridge, and you can buy it from August the 1st. You can buy it already if you are interested. And it's precisely about how we are changing our perception of biology from genes to cells. And it's really a discussion of how cells and cell biology is the way to understand the development of animals. It is a popular science book, and if you are interested, as I said, you can find it in many places on the web. I'm not gonna dwell on it, but I hope that you'll enjoy it if you read it, and I look forward to hear your comments. And all I can do is to thank the people, and looking at this slide makes me very, very nostalgic, that over many years in Cambridge and now in Barcelona have been part of the lab and have been part of the discoveries, the excitement, the discussion, the ideas, and also sometimes the frustrations, the pains that we all have shared their lives, because sometimes, as I said at the beginning, it's very difficult to distinguish life and science in our trade. But I can assure you that it's worth it, as long as we remain loyal to the principles that we have. And with that, all I can say is thank you, and look forward to your comments and your questions.
Thank you so much, Alfonso. That was a fantastic talk. We have a few questions in the Q&A section. So Jayant Maini has a couple of questions. He wants to know what is the role of cellular memory modules in cell fate decisions, and whether poised promoters and enhancers also play any role in transient states before cell fate decisions.
Let's hope that I can remember the two parts of the question. I mean, the issue of cell memory, it's a very interesting one. You know, I think many people will interpret that as epigenetics. That would be the thing. There is no question that all those things play a role, but we need to think, and I think the same, of course, enhancers, but we need to, at the moment, I think the challenge is how do cells access those memories and those enhancers, and how they coordinate the utilizations of that memory and across cell populations. This is not clear. You know, it's very clear. Think of the blood. The blood, to me, is a good example that I like to put in. I mean, let's start with a simpler system, which is the intestine. In the intestine, there is some homeostasis, right? There is a number of progenitor cells, there is a number of stem cells, and there is a number of differentiated cells in a very carefully established balance, okay? If that balance goes wrong, you get cancer, you get malfunction, okay? What keeps that balance? We don't even know there. But that's a number of 100 cells per crypt. But now go to the blood, where these proportions are maintained over thousands of cells. And any small imbalance in those proportions goes completely awry. This is not regulated by genes. This is regulated by properties that we need to learn. The gastroloids that we make, or the embryo models that other people made, like Nicolas particularly, you know, you get those cells, you get separated, they will do interesting things, but you bring them together, and they seem to acquire information from bringing them together. And Nicolas was one of the first ones in showing that. We have no idea of what that information is. And it's not just simply chemical. Obviously, the cells are picking up. So those molecular devices are very important. But I think the time now is not only to continue mapping them, but to start asking questions about how do cells access them and control them.
Okay, that's great. Ayushi has a question about how does a cell identify the right time for growth in individual cells? At the very initial phase, how do cells time their growth and division time? This is in the context of development.
Yeah, this has a very simple answer. We don't know. Go and learn. I like questions with short answers.
Okay. Meya Kumla wants to know, what is the significance of lethal genes in the context of development and genetic studies? Again, it might be a long answer.
No, lethal genes were very important historically. In fact, there is a very famous book called Lethal Factors in Development by Ernst Haddon many years ago in Germany. I mean, lethal genes once, in fact, it was the great vision of Nusslein-Volhard and Wischaus and Jürgens to realize that behind these things that they were called lethal mutations, they were patterns that were misconstrued, that were messed up. So lethal genes have been very important in understanding development because they were not just things that cause death. In fact, when you look at the screen of Nusslein-Volhard and Wischaus, you could have imagined that every mutant might have died in a particular way. What was interesting is that there were classes, and the minute there are classes, that tells you that there is a sort of some kind of mechanism behind it. So lethal gene, I'm not sure that that's not very important because we know that these things exist. I always say, and people don't understand, and I will leave it for people to think, these days, give me a screen that doesn't give you a mutant, and I will tell you that's an interesting screen, because a screen is always designed to give you mutants. Now, if you find one that doesn't give you a mutant, that's telling you something.
Fantastic. Priyanka has a very specific question about human development. She says, during the transition from day 3 to 4 post-fertilization, what is the factor that guides cells to convert into the inner cell mass and initiate further differentiation? I think the question also goes on to say that the composition of the cells at that stage is almost identical. So why do only a few of them go on to form the ICM?
Maybe I should let Saurabh answer that question, but I will speak over him. Because again, the answer is, I think there is some very nice work. My colleague Christian Schroeter, for example, and Katja Antoniakis, they are very interested in those questions. And I do think that those are, because of their simplicity, very good system to be answering this system. In fact, what Christian has shown is that these things will happen even in culture, these proportions, and I think there is a big opportunity there. I think what we have is to move a little bit away from thinking that genes are driving these processes and start thinking in different ways. And when people tell me, well, then how? Think about it. At the opening of the 20th century, people had many mysteries. We didn't know anything about genes. 30 years later, we understand life with genes. Now, what we have to understand is that genes do not explain everything, and start asking different kinds of questions, which are going to open very interesting avenues. And I want to stress that people in the audience are going to have fun if they do that. Otherwise, they're going to spend their life looking at single-cell RNA-seq data sets, which is not bad.
Aditya Parekh has an interesting question. It's a non-scientific question. He asks, I'm sure, Alfonso, in your long and illustrious journey, you may have seen some big experimental results not matching your hypothesis, which you were very sure of, and it may not have been easy to deal with them. What was your perspective on this, and have you encountered any? So,
I think there are two parts to that question. I mean, now we enter into the divan of the psychologists. As somebody said, La biologie dans le boudoir. It's a book by a French interesting scientist called Alain Prochance, which is like the biology goes to the psychoanalyst. I think, yes, I did have I've had two painful episodes. I mean, the first one, as I mentioned in passing, I'm not going to expand myself. When after 2 years in Chicago, I realized that my dream of understanding biology through physics was just premature. I mean, it was a bit disillusionment that I couldn't do mathematics. Looking back, I think I learned a lot in my years in Chicago, but it was hard to accept that what I wanted to do. Now I can see that maybe it was too early, but later I made, it was a mistake. Sometimes in the days of cloning genes, you could spend 2 years cloning a gene only to find that you hadn't cloned the gene that you wanted, and you had thrown, either you had to sit with the gene that you want. Those are very difficult things, but I think like life, I don't think a science is different from life in what I'm going to say. You have to stand up and walk on and move along. As I say, there is no, it is difficult. I think in those moments, friends help to have a good perspective. This is also sometimes, and because you're asking this question, sometimes you might have problems in your lab with your supervisor or your work. Your supervisor might not want to hear the problems with your work. That's why having other people outside. So you cope with those things through mentors, through people, through friends, but inevitably that will happen. I also think for those of you who are young PIs, the job of the PI is precisely, I tell the students when I say, well, we haven't talked for a while, and they say, I don't have anything to tell you. No, I say to them, this is when you have to talk to me. When you have something to tell me, I'm not interested. You can talk to your mates. We are there to help, because science is tough, and it's tough precisely in that aspect. That a lot of the time we are dealing with things that don't work, with things that we don't understand. I remember someone, I asked one of my group now, I don't know how to do that. Well, that's what you get paid for. So go and learn, okay? This is what we don't understand sometimes, that our job is a very interesting job because we choose it, but we have to do it. It's a job.
That's a great response. Vignesh also has a question, and he wants to know, what new skills do you think that present-day students of biology should have in order to make a positive difference in the scientific world and in general to society?
I think, as I've mentioned before, I've interacted a lot with physicists, particularly physicists and mathematicians. And there is a very big difference between them and us. They have, these days, there is a lot of emphasis on transferable skills. And I do think that's something that they have. That is to say, one day they decide they don't want to study physics, they will go to a bank, they will go to an insurance company, they will go because they have skills. I think we don't. I think there are two things about biology today. One, very important, I always say, if you are below 25, you better learn to program immediately. If you are between 25 and 35, it's optional. If you're about 40, 35, forget it, you're useless. But I think that the learning computing, learning computer science, these days, ChatGPT, this is going to be very, very important. I'm encouraging people in the lab to try to understand that. You have to get into understanding. This is what I said about understanding the world we do. I mean, the world in which we live. You are not there, guys, to provide data for your boss to travel the world talking about your work. You are forging your future. And the future now lies in all these computing skills and computer skills. So I think also something that is happening in biology and that you guys also have to think about, I think I sensed this a while ago, but it's very obvious now. I think there is a dichotomy. If you want to work in genomics or if you want to work in cell biology. And those are two very different skills. I mean, 40 years ago, you could try to do both at the same time and combine them. Today, if you do cell biology, you need to understand genomics. But you cannot do genomics at the same time that you do cell biology. On the other hand, you know, if you do genomics, there is so much there that in principle you don't need to do anything. But I think this dichotomy is absolutely real, and some of you probably can see it in your working places. So choices again, you see, making choices.
All right. I think we're at the end of our session now. So if there are any more questions in the,
If anybody wants to reach out to me, I'll be very happy to discuss with them and answer questions online. I think they can find my email in the web, and I'll be very, very happy to do that. Saurabh.
Yeah, I had a question. Like I was always wondering, like fascinated about your gastrolite system. So maybe I missed it. You purposefully decided to make an in vitro system to study gastrulation, or you were aggregating cells and you saw them organizing in a way that they resemble gastrulation? And so
I'm going to be very shameless here. Read the story in the book. But anyway, it's a very small part of the book. No. That was by design. In fact, you'll be very interested to hear that we tried first to aggregate trophoblast stem cells, primitive stem cells, and we failed completely. So I thought, let's see what do embryonic stem cells do. We wanted a system to study how cells could govern their proportions in space. That was not an accident. What happened was an accident, but we were looking for that accident. And as happens sometimes in life, what came out was not what I expected. And I spent a lot of time trying to figure out what it means, okay? So it was not an accident. Okay.
It's something to...
yeah, but what came out was not what I expected, okay?
Great.
And I should say that that's the outcome of the best experiments. When you get the result, this is what happens many people. When the person comes in the lab and says, the experiment didn't work. What do you mean it didn't work? It didn't give you the result you want, or it didn't work? Because the surgeon cannot come and say, sorry, the patient died because the operation didn't work. I mean, that's a waste of money. I mean, you need, sometimes the experiment doesn't give you the result that you want. Then you have something to do. If experiment is well done, it will give you a result. But not doesn't work. Okay.
Great to hear that.
Okay.
All right. So apologies to those whose questions we could not bring up, but please, as Alfonso mentioned, you're free to reach out to him and email him. I'd like to thank Alfonso on behalf of all the participants and Saurabh and India Bioscience for a fantastic talk. It was really great. An absolute pleasure to have you.
Thank you, everybody, and you. And I hope to see you soon in person, Saurabh. We are not that far. It would be very nice to see you in India too, but Vienna is closer. Okay. Bye-bye.
Just a small announcement for the next series that of course we will make a big announcement later, but next talk of the series is on 26th of July, and that will be also fun because it's by Madeline Lancaster, that how she assembled brain organoids. So that would be quite interesting to listen to all the attendees as well. So great to see all of you in great number. See you soon.
Bye-bye.
Thank you very much. Bye-bye.